This is the lecture for Chapter 8, Microbial Genetics, Part 1. Nucleic acids are one of the 4 types of macromolecules that make up all living things, and the role of nucleic acids is to serve as the molecule of information for the cell. And the 2 most common types of nucleic acids that we talk about are DNA standing for deoxyribose nucleic acid, and also RNA, ribose nucleic acid. And like all macromolecules, DNA and RNA are composed of similar subunits that are linked together by covalent bonds. And those subunits, or monomers, are called nucleotides. And the basic structure for a nucleotide is this structure here. Every nucleotide has a phosphate group, a sugar group, and a nitrogenous base. And for DNA, there are 4 types of nitrogenous bases. 2 are pyrimidines, that's thymine and cytosine, and the other 2 are called purines, and that is adenine and guanine. To form a molecule of DNA, those monomers, the nucleotides, need to be linked together. And this is done by taking a nucleotide, here's one nucleotide, and adding it to the 3' end of a strand of nucleotides. And a covalent bond will be formed between the phosphate group of one nucleotide and the sugar of another nucleotide. So this is one strand of DNA, and here you have what we call a sugar phosphate backbone, because this long strand here is composed of repeating phosphate, sugar, phosphate, sugar, phosphate, sugar, and all the nitrogenous bases are sticking out in the center. So this is one strand of DNA, but you probably know that DNA is a double-stranded molecule. So this is what the double-stranded molecule looks like. This is one strand of DNA. This is a second strand of DNA. And again, this is one nucleotide here, covalently bonded to a second nucleotide. And this is your sugar phosphate backbone. And the nitrogenous bases are sticking out in the center. And the second strand is going to line up with complementary base pairing. So thymine always pairs with adenine, and guanine always pairs with cytosine. And so that is the complementary base pairing. And these 2 strands of DNA are held together by hydrogen bonds. And that's a very important aspect of DNA, because hydrogen bonds are relatively weak bonds, so it's easy to break a few of them. But when you get a lot of them together, you get a very strong force. So while double-stranded DNA holds together very strongly, it is relatively easy for enzymes to pull apart 2 strands of DNA in small areas. Also, you may know that DNA is a double-stranded helix. And so here is the strand again, 2 strands of DNA. And then in water, the whole thing twists to form a helix. Now, another thing to point out to you is that the sugar phosphate backbones are anti-parallel. So each backbone has a 3' end and a 5' end, and that refers to the carbon that is closest to the end. So in this structure on a 5' end, this 5' carbon is the furthest at the top, and over here a 3' carbon is closest to the bottom there. So a strand of DNA has a polarity. It has a 5' end and a 3' end, and when you put together 2 strands of DNA with complementary base pairing, they will be anti-parallel. On one side it will be 3' here, and across it will be 5'. And the other side will be the opposite. 5' lines up across from 3'. RNA is very similar to DNA, but there are a couple of differences. First is in the type of sugar. So RNA stands for ribonucleic acid, so the sugar is a ribose, whereas DNA is deoxyribose. So there's one less oxygen in the sugar in DNA. Another thing about RNA is that it is single-stranded. So all RNA molecules are single-stranded, whereas DNA is double-stranded. And the last difference is that instead of thymine, RNA has uracil as one of the nitrogenous bases. But otherwise, it also has cytosine, adenine, and guanine. So the uracil is going to take the place of thymine during complementary base pairing. One of the major processes that involves DNA is DNA replication, and this is part of cell division. DNA replication is where the cell makes a complete copy of its genome so that both daughter cells will have a full set of genes and will be functional cells after cell division. So this process only occurs right before cell division. So if you have a cell that is dividing frequently, such as hematopoietic stem cells, those cells will be going through DNA replication fairly frequently. If you have other cells that hardly ever divide or never divide, like a neuron, neurons will hardly ever go through DNA replication, because if a cell is not going to divide into two daughter cells, there's no need for the cell to create an exact copy of its genome. In a eukaryotic chromosome, this is an example of a eukaryotic chromosome because it is a linear chromosome, there are sequences in the DNA that are called origins of replication, or sometimes they're referred to as ORI, O-R-I. And the origin of replication, this is where the DNA replication will begin. And in a eukaryotic chromosome, because they're so long, there are multiple origins of replication. And when DNA replication begins, those hydrogen bonds that held the two complementary strands together will be broken, and that will create a replication bubble. And those bubbles will continue to expand until they go through the whole chromosome, and you end up with two identical DNA molecules. And each DNA molecule is made of an original parental strand of DNA and then a newly synthesized strand of DNA. And this type of replication is called semi-conservative. And it's called semi-conservative because the original DNA molecule is partially conserved. Each daughter molecule of DNA has one original strand and one newly synthesized strand. Also during DNA replication, enzymes can only add nucleotides to the 3' end. So the enzymes can only add to a particular end of the DNA. So when you unzip the DNA, one side is called the leading strand because the enzymes bind and they can continually use complementary base pairing to form the complementary strand. The other is called the discontinuous strand because they can only add in the 3' direction, so they have to wait for the strand to open. So this creates what we call Okazaki fragments, so fragments of replicated DNA, which later, after the whole strand is finished, you need to use an enzyme called ligase to form covalent bonds between those Okazaki fragments. When looking at DNA replication in more detail, you would want to look at the replication fork. And this shows you the parental strand of DNA as it goes through the process of being replicated into two identical daughter strands. And in this process, there are lots of different proteins that are involved, such as DNA gyrase, helicase, and primase, single-stranded binding proteins, and so on. But there are only two proteins that I really want you to know. The first is DNA polymerase, and DNA polymerase is this big green enzyme here and here and here. And DNA polymerase is the enzyme that makes the complementary strand of DNA. And so if you read this name backwards, it basically tells you that ACE means it's an enzyme that makes a polymer of DNA. So DNA polymerase is the enzyme that reads the template strand, so the template strand, and then matches the appropriate nucleotides for complementary base pairing. And the other enzyme I want you to know is DNA ligase, which I mentioned before. DNA ligase is going to create those covalent bonds in the DNA backbone to link the Okazaki fragments. So DNA ligase ligates together, sort of glues together those covalent bonds in the fragments that are made in that lagging strand or that discontinuous strand. So this would be the lagging strand and this would be the leading strand. Another thing you should notice is that it's also required to have a primer. A primer is a short sequence of nucleotides that the DNA polymerase recognizes. So for the DNA polymerase to actually function, you need a little bit of a primer to get it to start replicating the template strand. Prokaryotic DNA replication is a little bit different than eukaryotic replication, and that's mainly because of the shape of the chromosome. Prokaryotic chromosomes, if you remember, are circular. So they are circular chromosomes as opposed to eukaryotic chromosomes, which are linear. The other thing about the prokaryotic chromosome is it has one origin of replication. So one sequence of DNA that tells the enzymes where to start replicating the chromosome. And the thing about a circular chromosome is that if you start copying it on one side, then because it's circular, if you don't tell the enzymes where to stop, they're going to continue going round and round and round. So also on the prokaryotic chromosome, you have another sequence called the termination of replication. So that is going to tell the enzymes where to stop. On a eukaryotic chromosome, you don't need that because it's linear. So if you start replicating in one spot, at the origin of replication, you continue till you get to the end, and then getting to the end tells you to stop at that point. So for prokaryotic DNA replication, you need an origin of replication and a termination of replication. And of course, replication at the origin of replication, it will start at that one spot. The two strands of parental DNA will be unwound and unzipped. Those hydrogen bonds are broken. And then DNA replication will occur in both directions until you get to the termination of replication. So you get to the termination of replication, and that's where the enzymes will stop. And you will end up with your two separate identical copies of the prokaryotic genome. The second important process that involves nucleic acids is called gene expression. And this is sometimes referred to as the central dogma of biology because it's sort of the central idea on which all aspects of biology rest. And this idea starts with DNA. And DNA, as I mentioned before, is the molecule of information. And when we're referring to it in terms of the information, we refer to those sequences of DNA as genes. And those genes encode all sorts of information, but most of the time they encode information for the cell to make certain proteins. So even though there are genes that encode other types of information, for this class, anytime I say gene, I want you to think in terms of a protein product. And within those protein products, the vast majority of proteins that are produced by a cell's genes are enzymes. And you should know that enzymes are biological catalysts that catalyze different types of biochemical reactions. And those reactions that a cell can produce or can perform will give that cell certain characteristics. So this is the idea behind gene expression, or the central dogma of biology, that the genes that an organism has directly relates to specific characteristics that that organism has. And it's all because the genes, or the vast majority of genes, encode for proteins. And again, most of those proteins are enzymes, which will catalyze different types of biochemical reactions. And it's those reactions which give the cell specific characteristics. And cells need to have biochemical reactions occurring all the time to be considered alive. They're metabolizing all the time. So this idea of gene expression, this is occurring all the time in living cells because the cells need a constant supply of enzymes for that constant state of metabolism they have to be in. And this will also be important when you're identifying an unknown bacteria because you're looking at characteristics of different types of bacteria. These characteristics relate to what sort of metabolism they have. And that relates back to the enzymes, back to the genes that they have that define them as a particular member of a species. The overall process for gene expression is described in this figure. And remember, as I mentioned before, gene expression, which is occurring all the time in every living cell, involves taking the information that's in DNA and creating the protein product. And remember, the protein is probably some sort of enzyme which gives the cell a characteristic. But this process, you don't go straight from DNA to protein. Instead, you need to take your template strand, your coding strand of DNA, and create a copy in RNA. And that process is called transcription. Once you have that messenger RNA, then you use that information to create the protein, and that is called translation. Transcription and translation are very closely related in terms of words and spelling, but they mean very different things. So I'll try to help you remember the difference between transcription and translation. So for transcript, if you make a transcript of a TV program, what you're doing is you're taking the spoken English and converting it to written English. So you're working in the same language with the same alphabet. DNA and RNA are the same molecular language. So you're using the same building blocks, the nucleotides, to build similar molecules. So an example could be if in DNA I have two words, big kid, and if I want to transcribe it using the same alphabet but in a slightly different form, I could use messy cursive and write big kid. So that would be transcription. Translation, on the other hand, when we use translation in regular conversation, we're talking about taking information in one language and translating it into another language. So when you look at RNA and protein, they are two totally different molecular languages. The monomers that make up the RNA are nucleotides. The monomers that make up protein are amino acids. And they have two completely different structures. So if I were to translate these two words, big kid, into another language, like Japanese, let's say, big is one character like this, and kid is another character like that. So transcription is when you're working within the same language, same molecular language. Translation is when you are working with two different molecular languages, when you're going from RNA to protein. Transcription is the first stage in gene expression, and transcription is where the information that is in the DNA is copied into RNA. This is an example of transcription. And the first thing I want to point out to you is that the promoter is a sequence of DNA that indicates where the gene begins. So the promoter is going to signal to the enzyme where to start transcription. And the enzyme that is involved in transcription is called RNA polymerase. And RNA polymerase, very similar to DNA polymerase, RNA polymerase is an enzyme that makes a polymer of RNA. So what the RNA polymerase is going to do is it's going to read the template strand. So it will read the nucleotides on the template strand, and then it's going to match with complementary base pairing the RNA nucleotides. So it's going to create a strand of RNA off of the template DNA strand. And that RNA strand is going to be complementary to the template strand. So if on the DNA you have a template, on the DNA template you have an adenine, the RNA polymerase will match a uracil. If you have a cytosine on the DNA, it will match a guanine. If you have a thymine on the DNA, it will match an adenine. So again, this RNA that's being produced is complementary to the DNA. And again, remember RNA has uracil in the place of thymine. So once the RNA polymerase recognizes the promoter, it will start unwinding and unzipping the DNA and creating that messenger RNA, which is a complementary strand from the template strand. And that RNA polymerase will keep sliding along the DNA, transcribing the DNA, creating the mRNA, until that RNA polymerase gets to a special sequence of DNA called the terminator. And the terminator is going to tell that RNA polymerase to stop transcribing the DNA. So the terminator basically tells the enzyme, the RNA polymerase, that that is the end of the gene. And remember, the gene is the coding sequence of DNA. The gene provides the information to make a protein. So a gene is the area on the chromosome between the promoter and the terminator. And that is the area that will be transcribed into the mRNA. So this blue strand here is the mRNA. So transcription. Transcription is taking the information from the template DNA strand and making a complementary strand in RNA. So you can see as you match it together, adenine with complementary binds to uracil, cytosine to guanine, and so on. The second stage is translation, and translation now is a little bit more complicated than transcription. Because remember, in translation, you are taking information that's in one form, single-stranded RNA, and you are making an amino acid sequence, a protein. For translation, one of the most important things that is needed is the genetic code. And this is one example of the genetic code. This is the classic example of the genetic code. And this is the same information in a slightly different form. But for the genetic code, it correlates codons, and a codon is 3 RNA nucleotides. So this example is U, U, U. And it correlates that codon to an amino acid. And remember, the amino acids are the building blocks for proteins. So the codons are found in the messenger RNA, and the amino acids will be linked together in correct order to form the protein. Now, most of these codons, there are 64 different codons because there are 64 different possible combinations of 4 nucleotides from the RNA. Most of the codons encode for different types of amino acids. But there are regulatory codons. The first one is AUG, adenine, uracil, guanine, and this is the start codon. And it will tell the ribosome where to start translation. There are also stop codons, so 3 stop codons, and those are UAA, UAG, and UGA, and this will tell the ribosome where to stop translation. Now, the genetic code is a universal code. Universal meaning that all living things on Earth use exactly the same genetic code. So the start codon, AUG, indicates the beginning or where translation should begin. In human cells, in bacterial cells, in plant cells, in all kinds of cells. So this is a universal genetic code. Transfer RNAs are also really important for translation. And transfer RNAs are often referred to as tRNAs. And the purpose of the transfer RNA or the tRNA is to match the appropriate amino acid to the codon on the mRNA. And this shows the structure of tRNAs in 3 different forms. And basically a tRNA is a single strand of RNA that kind of binds to itself with hydrogen bonds, forming this cloverleaf structure. But the 2 really important parts of the tRNA that you need to know is first the amino acid attachment site. So the amino acid attachment site, and this is where an amino acid will be attached. The other part is the anticodon. So the anticodon, this is going to be complementary to the codon on the mRNA. So if this anticodon is AAG, that means it can complementary bind to the codon on the mRNA that is UUC. So this is the tRNA that's going to bind to the codon UUC. And the amino acid that is encoded by UUC, if you look on the genetic code, is phenylalanine, PHE, phenylalanine. So this tRNA is specific for the codon UUC, and again through the genetic code, the appropriate amino acid that matches with UUC is phenylalanine. So in a cell, there are 64 different types of transfer RNA, 1 for each codon. And remember, this anticodon is complementary to the codon on the mRNA. Ribosomes are the enzymatic complexes that perform translation. And ribosomes are fairly large molecules, and they are composed of protein and RNA. And the RNA that you find in the ribosome structure is called rRNA, so little rRNA. And the ribosomes have 2 parts. They have a large subunit and a small subunit. And the small subunit is what recognizes and binds to the mRNA. So once you have performed transcription and have an mRNA, it's the small ribosomal subunit that will recognize and bind to that RNA. Then the large subunit will come in, and the large subunit has sites for tRNAs. And these 3 sites are called the E site, the P site, and the A site. And that is where the tRNAs will bind. And the A site is the attachment site, P is peptide site, and E is eject site. When a ribosome actually performs translation, it is basically doing 3 things. The first thing it has to do is read the codon on the RNA. Codon. So this is the RNA here, so it's going to bind to the RNA and it needs to read the codons on the RNA. And so remember, the codon are the 3 nucleotides found in the RNA. Once it reads the codon in the RNA, the second thing it's going to do is match the appropriate tRNA to the codon. And the way it matches the appropriate tRNA is by that complementary base pairing. So if this is UGG, then the matching tRNA would be ACC, a complementary RNA sequence. And so remember, on the tRNA, this is called the anticodon. Once it matches the appropriate tRNA, then it is going to form a peptide bond. Peptide bond. And the peptide bond is what links amino acids together. So that is the peptide bond there. So the way translation works is that the RNA, or the ribosome, will bind to the mRNA, it will read the codon and match the appropriate tRNA. And that tRNA had an amino acid bound to it. So that was a tryptophan. Then once it has matched the appropriate tRNA, then it's going to shift over, read the next codon, UUU, match the appropriate tRNA, which will have the anticodon AAA, and it will form a peptide bond to those 2 amino acids. And once it does that, it will shift over, read the next codon, which is GGC. It will find the appropriate tRNA, which would be CCG, the anticodon CCG. It will bind here, and then it will form a peptide bond to that amino acid. And it continues to do that, so the ribosome will continue sliding along the mRNA, reading the codons, matching the appropriate tRNAs, and forming a peptide bond. And that's how you create the protein from an mRNA. Every student who takes the biology class should be able to use the central dogma to determine a protein product of a gene. So because of this, I'm going to give you a practice problem. And the practice problem is, what protein product is formed from the following sequence? So this is the following sequence that I want you to find the protein product. So the first thing you have to do is determine what kind of molecule is this. And because it has thymines in it, that should tell you that this is a sequence of DNA. So since you know this is a sequence of DNA and you need to find the protein product, that means you have to transcribe it first into mRNA and then translate it into the protein. So to transcribe it into RNA, you would assume this is the template strand and you want to make a complementary strand in RNA. So that means wherever you have a cytosine, the RNA polymerase would match a guanine, so guanine. Then wherever there is a thymine, that would be matched to an adenine, adenine to a uracil, because remember this is RNA. And cytosine to a guanine, thymine to an adenine, adenine to a uracil, and a guanine to a cytosine, and so on. So C would match to G, A to U, U, C, 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 U, G, adenine, A, cytosine. So that is your mRNA. So that is your mRNA. Now what you're going to have to do is translate into the protein. And for this, you need the genetic code. So here's the genetic code that you can use. Now the first thing you want to do is find that start codon. So you would slide along the mRNA until you find an AUG, start codon, and here it is. And that start codon is going to set your reading frame. So what that means is that every 3 nucleotides on your RNA is a codon. So that's your reading frame that you've set by finding the AUG. And once you find all your codons, then all you have to do is look at the genetic code and find the corresponding amino acid. So AUG is the start codon. It also encodes for methionine. Then AUC is isoleucine. Then GUU is valine. And then CCC is proline. Then UGA is a stop codon. So when you get to a stop codon, you stop. So for this problem, what protein product is formed from the following sequence? The sequence I gave you was DNA. You needed to transcribe it first, and when you transcribe it, you use complementary base pairing. Once you have formed that mRNA, then you need to find the start codon, because the start codon is going to set your reading frame. So it sets your reading frame, and then all you have to do for the protein is to look up each codon and determine which amino acid is encoded by each codon. And this is the protein that is encoded by that DNA sequence. Eukaryotic gene expression can be relatively complicated because the 2 different processes, transcription and translation, occur in 2 different parts of the cell. So remember, a eukaryotic cell is one that has organelles. It has a nucleus, and the nucleus is where the DNA is kept to keep it protected. And remember, DNA never leaves the nucleus. So that means that any process involving DNA, like transcription, needs to occur in the nucleus. Ribosomes, on the other hand, are out in the cytoplasm. So once transcription is performed, then that messenger RNA needs to be transported out of the nucleus where it can interact with the ribosomes and be translated into a protein. And in order to get the RNA transcript out of the nucleus, it has to go through RNA processing. So that's where the exons are spliced out, and the RNA is further processed with adding a poly A tail and a cap at the front end. So this process can take kind of a long time. Transcription is occurring in the nucleus. The RNA has to be processed. It has to be transported out, come in contact with the ribosome, and then translation can take place. Prokaryotic gene expression, on the other hand, can happen very quickly. And that is because of the structure of the prokaryotic cell, basically meaning that that prokaryotic cell does not have a nucleus. So this is an example of a prokaryotic cell where the prokaryotic genome, the circular genome, is out in the cytoplasm with the ribosomes. So that represents a ribosome. So because they're not divided and they're all together, that means as soon as transcription starts occurring, so this is the transcript, the purple RNA, as soon as it's formed, ribosomes can bind and start creating the protein. So that's the protein there. So ribosomes can start translating the mRNA before the mRNA is even finished being formed. And so that means that prokaryotic gene expression can occur a lot faster than eukaryotic gene expression, which means that the prokaryotes can respond to changes in nutrients, changes in the environment much faster than a eukaryotic cell.